MekongDNA is a 3-year project from 2021 to 2023 that aims to apply environmental DNA or eDNA methods to explore the whole microbial communities or microbiome in the Lonsang Mekong River. Our goal is to use eDNA as a tool for biomonitoring in terms of biodiversity dynamics, ecological changes, pollution detection, or environmental effects on the river, and to develop a natural bioindicator for the Lonsang Mekong ecosystem. To get a comprehensive picture of biodiversity in the Mekong River, currently we have collected water and sediments at the 7 hydrological stations along the river in 3 countries. From the upstream region of the Mekong River in China, the middle region of the river in Thailand, and the downstream region of the river in Vietnam. Local officials at the hydrological stations from the Department of Water Resources helped collect water and sediment samples from the river for the project. To obtain eDNA from the water samples, each sample is filtered through polyether sulfone membranes. At the lab, metagenomic DNA is then extracted from the samples on the filter membranes. This eDNA will be amplified by PCR to indicate microbial species in the samples. Then, the PCR products will be sequenced by next-generation sequencing. After getting the sequencing results, the DNA metabarcode sequence data will be processed and analyzed to determine the diversity and composition of microbial species in the river. We explore the taxonomic composition and diversity of microbial communities in the river, assess the community structure and the compositional heterogeneity across different sites and habitats. Temporal and spatial analysis of microbial communities would help us understand the response of microorganisms to changes in the Mekong River environment. The physicochemical characteristics of the collected water and sediment samples are also analyzed to see their effects on the microbial diversity in the river. The relationship between physicochemical hydrological factors and microbes are compared to uncover potential bioindicators.
Besides the eDNA approach, we also use several novel culture-based techniques to isolate and identify microbes in the Mekong rivers that are rare or difficult to grow. In particular, the microfluidic streak plate is a droplet microfluidic-based approach that we use to isolate microbial cells individually, and it let us understand microbial consortia at a single-cell level. iChip and IC-tube are techniques that isolate individual microbes by growing the microbes under conditions that simulate the real environment in which they live.
With these methods, we have already been successful in isolating some interesting microbial strains from the Mekong. Both the culture-independent and culture-dependent approaches let us expand the information on freshwater microbes. The website for the project is set up and will be used as a platform for data dissemination and communications among members, as well as for public communication and awareness. The project website is www.mekongdna.org. Moreover, microbial DNA data can also be accessed and explored with analytical and visualization tools available at the Amibase website at www.amibase.org. Besides, we are also interested in capacity building through Citizen Science Program, which introduces local students and volunteers to eDNA experiences in a workshop series. Due to the COVID-19 pandemic, our first workshop was organized online on September 16, 2021. During January to November 2022, when the situation was better, we had conducted 4 on-site workshops, collaborating with our allied universities in North and Northeastern provinces of Thailand. The last workshop, our biggest one, was held on 28 to November 29, 2022 in Bangkok, in collaboration with the ASEAN Satellite Training Course of WDCM Global Training Course and the GBIF Project Microbial Metabarcode Database of Mekong River. In total, our Citizen Science program has trained 317 individuals. In the end, our project so far has completed a year-round sampling of eDNA from water and sediment of the Mekong River in year 2021 to 2022. The metabarcoding analysis of those eDNA samples showed bacterial diversity in the Mekong River, which comprised over 100,000 OTUs of bacteria. With bioinformatic tools, we can assess the relationship between physicochemical and hydrological factors and microbial communities in Mekong, which could help identify the potential bioindicators. Moreover, thanks to our Citizen Science program and the project website, our project has gained a lot of public awareness, even exceeding our expectations. So we hope our result will benefit all stakeholders in water resource management and in policy making in the long run. Thank you for watching. See y'all next time.